All right, we are on to chapter three now. In this chapter, we're basically going to talk a lot about the cell and everything that's in the cell, all of the organelles, the nucleus. We're going to talk about different forms of transportation between the intracellular and extracellular world, as well as cellular reproduction. So to start off, and we've said this before in class, a cell is the smallest unit of life. Yeah. The rest of what is written on here is kind of just more for your contextual information, right? We know that cells interact to form tissues. Tissues then form organs. Organs form organ systems that leads to our organism. The human body consists of about 30 trillion cells that have different size, shape, function. And the difference in shape and arrangement of the cells is what makes the function of a cell different, and the different functions of the cell possible. So, for example, a nerve cell has long extensions that conduct electrical impulses, while epithelial cells are flat, thin, densely packed. They create our skin layers. So, how a cell looks, its morphology, is going to inform what it's actually doing, its physiology. And that's kind of what we talked about all the way back in our definition of anatomy and physiology. And the big thing here is that genes control the cell's actions and responses. A gene is a piece of DNA that programs for function. Yeah. So just appreciate here the different types of cells. At the top, we have a nerve cell. At the bottom left, we have skin cells. And the bottom right, we have muscle cells. A composite cell is simply any cell that has different specialized structures within it. So by having organelles, you are a composite cell. All composite cells have different structures, but not every composite cell has the same different structures. The big components of a cell that I want you guys to know is the nucleus, the cytoplasm, and the cell membrane. Nucleus, very simple, contains the DNA, the genetic material that directs the cell's activities. Yeah, DNA programs what the cell does. It's in the nucleus. The cytoplasm is everything outside of the nucleus that is the cell. So it's the organelles and the fluid that the organelles float in. The majority of the cell is the cytoplasm. Okay. The cell membrane is just the boundary that encloses the cell. Think of it as the skin, right? It's the outer border of the cell. Within the cytoplasm are our organelles, which literally stand for little organs that perform specific functions to help the cell survive. And the fluid that the organelle is in is called the cytosol. So sometimes we think of the cytoplasm as the fluid, but the cytoplasm is the fluid called cytosol and the organelles. The actual liquid substance alone is just simply called cytosol. Here is a picture of a composite cell, so we can see the cell membrane that makes up the border, and we can see all those very different looking organelles on the inside. The fluid that exists between them is the cytosol, so the organelles and the cytosol make up the cytoplasm, and then at the center there, the big blue orb is our nucleus. Starting with cell membrane. So also called the plasma membrane. Remember from chapter 2, we talked about the phospholipids. This is what composes the membrane. Yeah, phospholipids. It's the boundary that contains the cell's contents. Again, it's kind of like the skin of the cell. Regulates the movement of substances in and out, and participates in signal transduction and communication with other cells. So I want you to know the cell membrane is the skin of the cell. It's composed of phospholipids, and the cell membrane regulates the movement of things in and out of a cell, as well as its communication with other cells. Yeah, so it's generally extremely thin, flexible, and elastic. I want you to know this: a cell membrane is selectively permeable. All that means is that the cell decides what can get in and what can get out. Yeah. And it does that by having a bunch of different surface features that are designed to determine whether something can get in or get out. Some things can just get in and out freely of a cell. Most things need to be selected for getting in and out. It's how the cell makes sure it controls what is there and what isn't. So the cell membrane, like I said, is composed of lipids, specifically a phospholipid. Okay, the membrane consists of a double layer or a bilayer of phospholipids. I talked about in chapter 2 how they are lined up tail to tail and the heads are facing out because remember the phospho head is hydrophilic, so it likes the water, so it faces the fluid either inside the cell or outside the cell. And the fatty acid tails are hydrophobic. They do not like the water. So when you align two phospholipids with the tails pointing towards each other and the heads pointing out, the tails create this impermeable water tight layer so that fluids can't get through. Okay. The lipid soluble molecules, that is to say, a molecule that's outside and inside of a cell that can pass through fats easily, do just that. They pass through the cell membrane without any help. So, respiratory gases, steroidal hormones, these are things that are soluble in fat and can freely pass through fat layers. So, the cell membrane doesn't need to do anything special to let them in and out, they just pass freely. Otherwise, the cell membrane is impermeable to water soluble molecules. Okay. Additionally, I want you to know that the phospholipid bilayer that composes the cell membrane, because it's very flexible and thin, it's not very stable. So to help create stability, cholesterol molecules are embedded in it to give it more of a harder shape. Now the cell membrane is also has embedded in it proteins. Yeah. So proteins are classified by their relationship to the bilayer of phospholipids. If the protein spans the width of the bilayer and might even protrude on one or both sides, it's called an integral protein. If it protrudes on both sides, it is a transmembrane protein. So it is an integral protein that protrudes on both sides specifically. And if it's just a protein that sits on the membrane, either just on the out layer or on the inner layer, then it's just called a peripheral protein. Yeah. So integral, if it's embedded into the layer and protrudes on at least one side. It's a transmembrane protein if it protrudes on both sides, basically creating a channel all the way through the cell membrane. And if it's just hanging out there on either the inner or outer layer, it's just called a peripheral protein. Don't worry about what the membranous proteins look like. Just know that some of these proteins function as receptors or growth factors, okay? The receptors are going to be things that detect stuff on the inside or outside of the cell and then help it move across it. And then growth factors are responsible for helping build and repair cell membrane. Additionally, those transmembranal proteins are generally going to help carry things across, often things like ions, and in that case, they're just called ion channels. So this is about how things move in and out of the cell. Proteins are the thing that does it when it needs something special. That's what I really want you to take away from that. Proteins are not the only thing that are on the phospholipid bilayer. There are also carbohydrates. So some of these proteins have carbohydrates attached to them that we call glycoproteins. Glyco, think glucose, think glycogen. It's another word for carbohydrate. So proteins that have carbohydrates on it are called glycoproteins, sugar proteins. Glycoproteins specifically help a cell identify that it is a part of the organism. So it detects the self of the cell, and that way our immune system doesn't go around attacking our own cells. So every cell has glycoproteins in it that say, hey, I'm a part of the body, don't attack me. There are also proteins that protrude into the inner cell and anchor basically these supporting rods and tubules. They form what is called the cytoskeleton. So imagine you have the phospholipid bilayer, that's the skin. Well, proteins under the skin inside that phospholipid bilayer also help create a rigid structure so that that skin doesn't bend too much. That's called your cytoskeleton, okay? So we have steroids embedded in the phospholipid bilayer to help make that skin more tough. We also have these proteins beneath the phospholipid bilayer that form the cytoskeleton. Cyto meaning cell, skeleton, okay? So the membrane is the skin. The proteins underneath that form a hard supportive structure of rods and tubes. That's the cytoskeleton. And then steroids are embedded in the skin just to help give it some additional rigidity. Membrane proteins called cellular adhesion molecules. Just know this term. It helps determine a cell's interaction with other cells. It's how it communicates. Cell adhesion molecules just determine how one cell communicates with another cell. Yeah, it's often abbreviated as CAMS. So here's a picture. So we have these phospholipids, right? So this is our phosphorus head. We have the fatty acid tails, and it's not illustrated here, but remember all lipids have a glycerol group as well. So when they align themselves tail to tail, this head likes water. It's hydrophilic, so it faces towards the fluid outside of the cell, the extracellular fluid. This side focuses its orientation towards the intracellular fluid. These tails do not allow fluids to freely cross. It is impermeable to water-soluble molecules, so it doesn't allow molecules that like water to just freely flow in and out of the cell. Down here we have our cytoskeleton. Yeah, these are proteins that help give some rigidity to support the cell membrane so that it doesn't rupture easily. And here we have some steroids that, again, just are embedded in the cell membrane to give it added strength as well. Here we have our carbohydrate chains. So we can see a little bit of a protein there. This is a glycoprotein. This carbohydrate chain is a signal to the rest of the body to say, hey, I am a cell for the body, so don't attack me. And then these are a bunch of other examples of different proteins. So this is going to be an integral protein. This is going to be a peripheral protein. This is going to be a transmembranous protein because it goes all the way through. So integral, it's embedded, but it only protrudes on one side. Peripheral, it just attaches on the outside. And then a transmembranous protein, sometimes also called a channel protein. This is your big letting things in and out. The whole cell membrane is selectively permeable. All of these elements work together to decide what can and cannot get in and out of the cell. All right, the cytoplasm. This makes up the largest volume, right, of the cell. It's mostly liquid, specifically called cytosol. And inside that cytosol, inside the cytosol are floating structures like the cytoskeleton and the organelles. An organelle performs a specific function of the cell. It helps make the cell operate, okay? They're the organs of the cell. First big one is ribosome. What I want you to know about ribosome, right? It is composed of RNA, ribonucleic acid ribosome. Okay, remember, all nucleic acids have a sugar, a phosphate, and a nitrogenous base. That sugar is going to be ribose, and so ribose makes up RNA, makes up ribosomes. Okay, the thing I want you to know here is that ribosomes freely float in the cytoplasm, but it also bonds to a special organelle called the endoplasmic reticulum. And what I want you to know about ribosomes is that they synthesize proteins, okay? Yes, there is some structural support that it provides, but what I want you to know is ribosomes made up of RNA synthesizes proteins. Some of them are floating around freely in the cytoplasm. Some of them are bonded to what's called the rough endoplasmic reticulum. Okay, let's talk about our first big organelle, the endoplasmic reticulum. Here's what I want you to know. The endoplasmic reticulum is the transport network of the cell. There are two portions of the endoplasmic reticulum. There's the rough portion and the smooth portion. The rough portion starts with an R, so does ribosome. The rough portion contains your ribosomes. So it produces proteins, okay? So it's a transport network for the cell. You produce your proteins in the rough endoplasmic reticulum. The smooth portion does not have ribosomes. It's more involved with lipid synthesis. So it builds fats, it absorbs fats, it metabolizes drugs. I want you to mostly know it has more to do with fats. So this is the production center. The endoplasmic reticulum produces proteins in the rough portion, which has ribosomes, and has functions related to fat synthesis and absorption in the smooth portion, which has no ribosomes. This is what it looks like. I'm not really going to ever show you a picture of this again, but you can see it under the microscope. Again, the parts here that don't have ribosomes are the smooth portions, and then here is the smooth, and then the part here is the rough portion, yeah? So here you can see the membrane of the endoplasmic reticulum, and all these little specks are all the ribosomes. Here is the actual cytoplasm. So that's the end of the organelle. Okay, vesicles, they're transport sacs. That's it. When you need to transport something from one organelle to another, it gets put into a membranous sac. And basically, it's a mini cell membrane made up of phospholipids, and it's just a little sac that wraps around the thing that you're transporting, and it travels it to somewhere else, okay? So the endoplasmic reticulum produces stuff. It's your production center. The rough ER produces proteins. The smooth ER either absorbs fats or produces fats. Once it's produced, a vesicle will form around the protein or the fat, and it will carry it to the Golgi apparatus. The Golgi apparatus, sometimes known as the Golgi body or the Golgi complex, modifies these proteins and fats. Okay, it modifies them so that they're fully constructed to do what they need to do. Once that modification happens, into another vesicle it goes, and then it's either sent somewhere else in the cell where the protein or the fat is used, or it's pushed out of the cell to go to another cell or another part of the body where it's used. If it leaves the cell, it's called exocytosis. Exit the cell. Exocytosis. Okay? So again, the order of operations is the endoplasmic reticulum, sometimes known as the ER, that produces proteins or lipids, puts them in a vesicle, which is just a transportation sac, goes to the Golgi apparatus where it's modified so it's now a fully functioning protein and lipid, goes into another vesicle, either goes somewhere off inside the cell where it's used, or through exocytosis, it's pushed outside the cell to go somewhere else where it's used. This is your Golgi apparatus. Yeah. So here's an example of a vesicle. Okay. And it is traveling here. Oh, okay. It's actually showing something else. So here's the endoplasmic reticulum. We create the protein. We create the vesicle. It travels over to the Golgi apparatus, gets pulled into the Golgi apparatus, gets modified, put into another vesicle that breaks off. And in this example, it goes through exocytosis out of the cell membrane. The mitochondria. What do you think I want you to know about this? It's the powerhouse of the cell, okay? All I really want you to know is it is involved in cellular respiration. That is to say, it takes energy from the nutrients that we absorb and it produces ATP, right? So it takes the nutrients and uses it to produce adenosine triphosphate, okay? It is a double layered organelle. Okay, there is an outer layer and an inner layer. The inner membrane is folded up into what are called cristae. Yeah, and just know that the term is cristae. Yeah, it's a very active. If a cell is very active, it might have thousands of mitochondria. Now, what's unique about mitochondria, and we don't have to get into like the history of how the first cell was created, but the mitochondria has its own special DNA, so it's kind of like a cell living in a cell. And back in the primordial ooze of existence, the first cell that was ever formed is theorized to have been a mitochondria. And then at some point, another cell ate it, and then they started forming a relationship together where the mitochondria would produce energy, and then the larger cell around it would keep the mitochondria alive. Again, you don't have to know that. What I want you to know is the mitochondria has two layers. The inner layer is all folded up into cristae, and then the mitochondria produces ATP. So again, just pay attention to this picture. This is your mitochondria, outer layer, inner layer, all these little folds are cristae. Okay? It's the powerhouse of the cell, makes ATP. And again, all these membranes for all these organelles, they're all phospholipid bilayers. Okay? That's the outer membrane of the mitochondria. That's the inner layer of the mitochondria. All right, lysosomes and peroxisomes. These are organelles that are about digestion. Okay, these are your enzymes. These break things down. A lysosome, I want you to know, breaks down nutrients. Okay, it also deals with debris and broken parts of the cell. So if a part of the cell is damaged, a lysosome will go and break it down to get rid of it as it repairs itself. If there's random debris in the cell that needs to be gotten rid of, the lysosome does that. If there's nutrients in the cell, the lysosome breaks it down. Peroxisomes are very similar to lysosomes, except they break down fatty acid and hydrogen peroxide, peroxisome. So because it has to do with hydrogen peroxide, I want you to think detoxification of alcohol. Yeah, so a peroxisome is more to do with peroxides and alcohol. The lysosome is more to do with nutrients and debris, but they all break things down. Like I said, in the cytoplasm, we have our cytoskeleton. It's just inside of or proximal to that phospholipid bilayer. And it's made of three component parts: microfilaments, microtubules, and intermediate fibers. Microfilaments are a part of your microfibrils in muscle. So microfilaments have to do with the movement of the cell. And so I want you to think filament fiber. Microfilaments create microfibers in muscle. Muscles are active. So the protein that makes up a microfilament is actin. That's all I want you to know. Microfilaments are a type of cytoskeleton material made up of a protein called actin, and they form microfibrils in muscle. Microtubules are made up of a protein called tubulin, and they're involved in cell division. That's what I want you to know. Microtubules are made up of a protein called tubulin, and they create these long web-like things, or they are long web-like things that are important for cell division. Intermediate fibers exist. Riveting. That's really, there's a wide variance in how they function, so I'll probably never ask you a question about this. Know that they exist, but they're not microtubules, they're not microfilaments, okay? But they do play a role in the rigidity of the cytoskeleton. Yeah. Yeah, don't worry about any of those pictures. Well, I guess we can say this. Microtubules, obviously bigger. Yeah. Microfilaments, smaller, intermediate, somewhere in between. Okay. Centrosomes. So a centrosome, and we'll talk more about this when we get to cell division because they play a big role in it. They are two cylinders called centrioles. So a centrosome is a structure that is made up of two hollow cylinders called centrioles. Centrosome, centrioles. One centrosome has two centrioles, and it has to do with the distribution of chromosome during cell division. Okay? Not super important that you know that it's made up of 9 groups of microtubules, but again, A centrosome will have two centrioles, and those centrioles will spit out microtubules like a web that will, when we talk about cell division, basically pull the chromosome where they need to go. So they're kind of the taxi service, okay? So we link them into a category with the cilia and flagella because centrosomes have to do with how things move around the cell. And cilia and flagella are also structures related to movement. Cilia is little tiny hairs on the outside of a cell, and it is most important for moving things like fluids or other cells. They're little tiny hairs and they kind of move in a wave pattern. So if you think of watching a baseball game when everyone does the wave, that's how cilia moves. And it pushes fluid through things. It also moves other cells. So imagine the cilia that line the inside of your lungs is pushing stuff in and out of your lungs. If it's along the throat, it's helping to push things in and along the throat. Yeah. There are many, many, many little tiny hairs that have to do with moving fluids or whole cells. The flagella is unique. Cells that have a flagella have only one normally, and it's a long, thin tail that helps the cell move through space. The only cell in the human body that has a flagellum is the sperm, and it's the sperm tail. That's the flagellum. It's what allows the sperm to swim and try and find an egg. So the flagella is a tail, the cilia is tiny hairs. The centrosomes are structures that create little like web appendages, which are the microtubules. Again, don't worry about these kinds of pictures under the microscope. But see, this is a good picture. This is cilia, many, many, many little hairs that push things along the surface of the cell. And then, of course, here's sperm, here's the flagella. It's a modal structure to allow for propulsion. All right, the nucleus of the cell. We know most of this already. The nucleus contains the DNA, okay? Now, I want you to think of the nucleus as being a sort of cell within the cell. So, like the cell itself, the nucleus is surrounded by a double layer, phospholipid layer, and instead of calling it a cell membrane, we call it a nuclear envelope. The nuclear envelope, much like the cell membrane, has holes in it where things can travel, only instead of calling them integral, transmembranous, and peripheral proteins, we just call them nuclear pores. Okay? So the nucleus has the nuclear envelope enveloping it, and it has little holes where things like RNA and proteins can get in and out of the nucleus via nuclear pores. Like the cell itself, it contains a fluid inside. Instead of calling it the cytoplasm, we call it a nucleoplasm. Okay? Now, the nucleus also has a sort of nucleus called the nucleolus. It's a dense body inside the nucleus, except it stores proteins and RNA. It's where ribosomes are created. So the nucleus is where the DNA is held. It's wrapped in a nuclear envelope and has nuclear pores so things can get in and out. And the fluid inside the nucleus is the nucleoplasm. Additionally, we need a center for the RNA. Inside the nucleus, we also have a nucleolus. That is where RNA and protein is composed, and that is also where ribosomes are created. So chromatin. Chromatin is the loosely coiled fibers of protein and DNA found in the nucleus. So normally in the nucleus, your DNA is kind of just a big wadded up mess of fibers. We call that wadded mess a chromatin. Yeah. When we condense the chromatin into really tight packed big pieces of DNA, that's when we call it a chromosome. Yeah. So when the DNA is just naturally sitting in the cell doing its daily cell stuff, we call it chromatin. When it's getting ready to divide and form cellular reproduction, that DNA, that chromatin, condenses into tightly packed structures called chromosome. So again, Here is the nucleus. We have the nuclear membrane, which again, phospholipid bilayer, okay? Inner and outer layer. We have the nuclear pores for things to travel in and out of. All these little strands, that's the DNA. And since it's not packed up, it's called chromatin. If it was densely packed up, we would call that chromosomes. At the center of the nucleus, we have the nucleolus. Inside the nucleolus is where the proteins and the RNA sit, and that's where ribosomes are created. Ribosomes and RNA proteins, they then can travel out into the greater cell via the nuclear pores. I love this. I'm going to keep it here because everything we just talked about, this is a great synopsis. Yeah. If you know these two slides, you know what I was talking about. There's a couple of details that I added that I highlighted in the previous slides, but this pretty much gives you everything you need. So really know these two slides and you know your organelles. It sounds like a lot, but if you look at it like this, it really isn't a lot. Again, cell membrane, integrity of the outer portion of the cell. No, it's a phospholipid layer, yeah, bilayer. Ribosomes, it synthesizes proteins composed of RNA, yeah. If you know this stuff, this'll get you pretty far, and then just know the rest of the little highlighted stuff that I emphasized in the earlier slides. All right, how do things move in and out of the cell? So remember, the cell membrane is a selective barrier, which means it can control what passes through it. Mechanisms of movement through that barrier happen in two ways. It can be passive, meaning that no energy or ATP needs to be spent in order to do it, or it can be active, which means we have to spend ATP to do it. What's the difference between the two? If it's passive, it's obeying what we call a gradient. So if there's more on the outside than there is on the inside, passive things will drift into the cell to balance it out, to find homeostasis. If there's more on the inside and less on the outside, passive transport will allow it to just drift through the membrane and balance out that way. If it's active, it's our only method of going against the gradient. So if there's a lot on the outside and little on the inside, but it's stuff that we don't even want on the inside, we use ATP to commit active transport and push stuff from a low concentration to a high concentration to kick that stuff out of the cell or to pull it in. Yeah. So there's a couple examples of each. When we talk about our passive transport mechanisms, we have diffusion, facilitated diffusion, osmosis, and filtration. When we talk about our active transport mechanisms, we have simply active transport, endocytosis, and exocytosis. So let's talk about our first passive mechanism, diffusion. It's simply a movement down the concentration gradient, okay? It is a movement of molecules or ions in a liquid or a gas from a region of high concentration to low concentration. So like I said, if you have a lot of a substance outside of the cell, not much inside the cell, passive diffusion will allow those substances to simply drift right through the cell membrane into the cell. It's caused by random motions and collisions. So the particles that diffuse passively are going to be things so small that they just go right through the membrane. And so the rule of concentration will cause it to go from high concentration to low concentration. So it just freely passes right through the cell membrane. When we balance what's on the outside with what's on the inside, we call that diffusional equilibrium. That's when there's no more on the outside than there are on the inside. It's equal. And we make a special note that sometimes things are still entering and exiting, but it's doing so in such a way that the total number on the outside and inside still remains equal. So what kind of things passively diffuse through simple diffusion? Things like oxygen and carbon dioxide. Those molecules are so small. Oxygen is O2, carbon dioxide is CO2. Those molecules are so small they pass right through the phospholipid bilayer, the cell membrane, okay? So it travels into the blood, into the lungs, into all the different cells, and it just passively diffuses. If there's more oxygen on the outside than the inside, the oxygen travels in. If there's more, that causes the oxygen to be used for energy production, which creates the waste product, carbon dioxide. So now there's a lot of carbon dioxide on the inside, not the outside. What happens? Carbon dioxide leaves the cell. Yeah. So it occurs only if the cell membrane is permeable to the substance. So the things that perform simple diffusion have to be small enough to just naturally pass. Okay. An example to use in real life is placing sugar in a glass of water, right? You put sugar in a glass of water, that water is more concentrated outside of the sugar than inside the sugar. So it will passively diffuse into the sugar cube. That will cause the sugar cube to dissolve until now there is the same amount of sugar molecules all throughout the water. There's no area where there's more water and less sugar. It's all evenly balanced. That's passive simple diffusion. When we talk about a cell membrane, here's our cell membrane. We have water outside, but we have water on the outside where there's more water, and then we have a solute, a molecule on the inside that is small enough to cross the membrane, and it's more dense on the inside. So what happens over time, these molecules are going to leave, a little more water is going to come in until eventually it becomes homogeneous, and there's equal molecules inside and outside. There's equal water inside and outside. Yeah. When we talk about oxygen, here's our capillary, right? It contains blood that is high in oxygen and low in carbon dioxide. So this is a capillary. We now have a cell that needs the oxygen because oxygen is used to create energy. So there's a lot of oxygen here. There's not much oxygen here. We're going to passively simple diffuse through the cell membrane of the capillary into the cell membrane of whatever cell this is. We'll call it a muscle cell. And then that will be used to create energy. It will now create the waste product, CO2. Because there's a lot of CO2 in the cell and very little CO2 in the capillary, it will now push that CO2 through passive simple diffusion into the capillary. Yeah. And because oxygen and CO2 are so small that they can pass right through the barrier, it's simple diffusion. There's no energy required. Let's talk about facilitated diffusion. Same rules apply. We're going from high concentration to low concentration, but these are substances that can't freely cross the membrane. They need a little bit of help. Yeah. So it's a method of transporting a substance across the cell membrane using a membrane protein. So this is where our transmembranal, peripheral, and integral proteins matter. One type of it uses ion channels. That is a special channel that is attractive to ions with different charges to suck them into the cell. Again, it has to have a higher concentration of the ions on the outside to push them through into the inside. It still obeys the gradients, yeah? Substances transported by facilitated diffusion, for example, sugar, glucose, or proteins, amino acids, cannot pass through the phospholipid bilayer since they are water soluble or they're too big. For one reason or another, the selective permeability of the membrane won't let them in, so they need the help of a special carrier molecule. That carrier molecule is a protein of some sort that when the thing attaches to it, it says, okay, we want to bring this in, let's do it. As far as a rate reaction, the more carrier molecules you have, the more quickly you can do this. The fewer you have, the slower a process. So let's take a look at a couple examples. So here we have a high concentration of substances, but they're too big to get through the phospholipid bilayer, or maybe they're water soluble and it won't cross that hydrophobic tail. So we have an ion channel here as an example, and that might have a negative charge. These might have a positive charge. So they get attracted and spit right into the cell. Yeah, they get attracted to the outside and spit right in. Or maybe we have these special kind of going back here, carrier molecules, where we have a molecule and we have a protein, and the protein is designed with a special portion specifically to fit that molecule. That molecule, once it binds to it, turns on that protein so it closes the outside, opens the inside, and spits it out. Again, it doesn't require energy. The charge of the ion pushes it in, the shape of the molecule pushes it in, but they can't freely travel through the protein, the phospholipid bilayer. They need the help of these proteins. In this case, both of these proteins are transmembranal proteins. But because it needs the help of a protein, it has to be facilitated. It has to be helped out. Same rules, high gradient to low gradient is what's going to lead it. But it can't freely travel, so it's not simple. It needs the help of these embedded proteins. It's facilitated. Osmosis. So osmosis is its own special case of diffusion. It's simply the movement of water across a semi-permeable membrane from an area of greater concentration of water to an area of lower concentration. What makes osmosis annoying is we talk about it in the opposite sense. Instead of saying, hey, more water goes to less water, we talk about solutes. Remember, water is a solvent, and anything dissolved in it is a solute. So, where there is lower osmotic pressure, that means there is a lower concentration of solutes. So, when we're talking about osmosis, water moves across a selectively permeable membrane from an area of greater water concentration or lower osmotic pressure and lower concentration of solutes. Because imagine if you have a lot of water, you have less stuff in the water because it's mostly water. So we call that a low osmotic pressure, and it travels to a place of lower water concentration or higher osmotic pressure because there's a higher number of solutes. So just understand osmosis uses flipped language. It's still high concentration of water traveling to low concentration of water, but we flip the words and we say low osmotic pressure to high osmotic pressure. Osmotic pressure talks about the solutes, not the water, okay? So during osmosis, water moves from lower solute concentration or low osmotic pressure to higher solute concentration or high osmotic pressure. It's saying the same thing. If you have a low number of solutes, it means you have a lot of water, so high concentration. If you have a high number of solutes, you have less water, low concentration of water. It's going from high concentration of water to low concentration of water. But because osmosis has to be difficult, we talk about it from low osmotic pressure to high osmotic pressure, because osmotic pressure is defined by where the solutes are. If there's more solutes, it's high osmotic pressure, lower solutes, low osmotic pressure. So osmotic pressure is the pressure needed to lift the volume of water from greater concentration of impermeable solutes to greater osmotic pressure. So what does it look like? We have a lot of water and low solutes. So this is low osmotic pressure. We have little water and high number of solutes. This is called high osmotic pressure. We travel low osmotic pressure to high osmotic pressure. So B goes to A. And the reason we talk about osmotic pressure is because unlike facilitated and passive diffusion, where at the end everything is balanced out from a volume standpoint, because what we're balancing is the relationship between water and solutes, it doesn't actually have the same volume, but we can see there's the same volume of water to solutes here as there is over here, even though this is a different total volume. The pressure is the same. The ratio of water to solutes is equal here as it is here, even though it's a different total volume. That's why osmosis is a little weird. And because osmosis is weird, we talk about tonicity. So I don't want you to worry about all of the jargon on this page. What I want you to know is tonicity affects the size and the shape of a cell. If you have a substance that has the same osmotic pressure inside the cell and outside the cell, we call that isotonic. Iso meaning the same, tonic meaning tonicity, the same tonicity, isotonic. When that happens, water does not move in or out of the cell, and the size and shape of the cell remain the same. If you have higher osmotic pressure outside of the cell, okay, means a higher number of solutes, means a lower amount of water. It should make sense then that the water will leave the cell because there's more water in the cell than outside. We call this hypertonic, increased tone. This will cause the cell to shrink because there is higher osmotic pressure outside the cell, meaning there's less water. The water will rush out. When we talk about tone, we talk about pressure. So high pressure means high solutes, means low water. So hypertonic is more pressure causing the cell to shrink. On the flip side, when there is lower osmotic pressure on the outside, that means there's lower solutes and more water, which means the water wants to rush inside the cell. This causes the cell to gain water and swell. We call that hypotonic because there is lower osmotic pressure outside. So all I want you to know about tonicity is it is a result of osmosis. And if osmosis is not happening because everything's even, then the cell shape does not change. We don't rush water in or out of the cell. We call that isotonic. If I put a cell in an isotonic mixture, the cell will not change because the water inside the cell is the same as the water outside the cell. If I put it in a hypertonic solution, the cell will shrink because there's more pressure out of the cell. It crushes the cell, pushes the water out. If I go in hypotonic, meaning low tone, that means the pressure outside the cell is lower, so it's going to cause the water to swell inside the cell, and the cell will get bigger. This is an isotonic solution. We drop a red blood cell in it. It's isotonic, so the water doesn't flow in or out at any different rate. It's balanced. It's isotonic. The cell looks normal. If we go hypertonic, there's a lot of pressure on the outside, which means there's less water on the outside than there is the inside. The water rushes out and the cell shrinks. If we go hypotonic, that means that there's low pressure on the outside, so there is a low amount of solutes, which means there's a lot of water. The water rushes in and the cell swells. And in this example, it swells so much it actually explodes. All right, so that's diffusion, and that's facilitated in simple diffusion as well as osmosis. Let's talk about filtration. This is the process of forcing a molecule through the membrane due to the exertion of pressure. I want you to think about blood pressure, okay? Due to the action of the heart, it creates a pressure that pushes things through, right? Filtration is used by the body to produce tissue fluids from blood and plasma. Water and small solutes are sent through the wall of the capillaries to deliver the oxygen and nutrients to the cell, while large particles like plasma remain in the capillaries. So what does that look like? We have the blood vessel, okay, in this instance a capillary. We have pressure. The actual filtration that's happening is that as that pressure increases, it forces the smaller molecules out and it keeps the larger molecules in. Okay, so if I say filtration, I want you to think blood pressure. I want you to think the pressure pushing things, okay? Okay, that was our passive stuff. None of these things, filtration, osmosis, facilitated diffusion, simple diffusion, none of that requires energy. It happens naturally. Active mechanisms. Our first one is the active transport. It is the movement of a substance from the lower concentration to a higher concentration. So it goes against the gradient. In order to do that, we need energy to force it to happen, adenosine triphosphate. So active transport is moving a substance from low concentration to higher concentration with the help of energy. And the thing that does that are special proteins that we call carrier proteins or pumps. So these are not too different from the transmembranal proteins we talked about in facilitated diffusion, except because they go against the gradient, they require energy. Understand one of the common examples here is sodium and potassium, but calcium and protons also do this. Protons, or positive hydrogen ions, are important in the mitochondria. That traveling of the mitochondria through active transport is what helps us produce ATP in the mitochondria. Calcium is related to muscle contractions. Sodium and potassium are related to nerve conduction. You don't have to know that. What I want you to know is active transport use pumps. Specifically, they are carrier proteins called pumps. It uses ATP to force something to happen. So we have more potassium we have more potassium inside the cell than outside the cell. But we want even more potassium inside the cell. Additionally, we have more sodium out of the cell than inside the cell, but we want even less sodium inside the cell. So these carrier proteins called pumps will bind, right? It'll take 3 sodium and bind, it'll take 2 potassium and bind, and then using energy, it'll suck the potassium in and push the sodium out. I don't need you to know this exact example just to answer any curiosity. This is a nerve cell. Nerve cells conduct sodium and potassium by a 3:2 ratio when it conducts a nerve signal. You don't need to know that. What I want you to know is that this is active transport via a pump. It's going to take something and push it against its gradient by using energy. So things will bind to the carrier protein or pump, and then when energy comes along, it'll push it against its gradient. Endocytosis and exocytosis. Okay, so endocytosis is used for molecules that are too large to be transported by other means. And so, like facilitated diffusion, it needs help. However, unlike facilitated diffusion, it's going against the gradient. So it's called endocytosis because we're pulling something in. And we use vesicles to do that. Remember, that's just a membranous sac that transports things. The membranous sac is formed of a phospholipid bilayer. Exocytosis is about pushing things out of the cell. Okay, so in each instance, the thing that we're trying to get contacts the cell membrane, and then in endocytosis, it's carried into the cell. Exocytosis, it's pushed out of the cell. So endocytosis has 3 versions: pinocytosis, phagocytosis, and receptor-mediated endocytosis. Don't get in the weeds here. I want you to know one term for each of these. Pinocytosis is cell drinking. Fluid comes into contact with the cell membrane. It gets sucked into a vesicle and pulled in. Phagocytosis is cell eating. Solid particles touch the cell membrane, gets wrapped in a vesicle and pulled in. Receptor-mediated endocytosis uses ligands. And I'm just going to show you the picture so you can better understand what I mean by ligands. So, pino and phagocytosis. Here's the cell, water or fluid of some sort that the cell wants to drink, comes into contact, the cell membrane sucks it in, creates a little vesicle around it, and then boop, it's inside the cell, where now the fluid can be traveled wherever it needs to go. For phagocytosis, solid particles do the same thing. They touch the cell membrane, get sucked in, enclosed in a vesicle, and boop, there it is. It's inside the body, or inside the cell. Now, with phagocytosis, because it's a solid thing and not a component part, this is where our lysosome comes in. So once that cell eats the substance, lysosome, which remember, what are they used for? They're used for breaking down nutrients. They come along, they enter the vesicle, they digest the solid particle, and then the nutrients gets exited from the vesicle where it can be used in the cell. What's left over is the waste products, which guess what? Get pushed out through exocytosis. Yeah? So this is endocytosis of a solid structure. So that would be classified as phagocytosis. Combined with the lysosome, digested, the nutrients are sent around the cell to feed the cell, and the waste product is pushed out via exocytosis. Yeah, the vesicle bonds to the outer membrane, it opens up on the outside, and we boop, push it out. Receptor-mediated endocytosis. So this is when there are special receptors on the cell membrane specifically built to match the exact shape of the substance they're trying to pull in. So remember that facilitated diffusion example where there was that circle that bonded exactly to the protein shape right here? It's the exact same thing, but instead of a transmembranal protein, it's going to be this little thing called a receptor protein that binds to a ligand. A ligand is just any substance specifically designed to match the receptor protein. Once it binds, that triggers a reaction in the membrane to perform endocytosis. It pulls it in, creates a vesicle, and then travels it where it needs to go. Maybe a lysosome breaks it up, whatever happens next. So it's a type of endocytosis that requires a special receptor protein. So when we're talking about these three, pinocytosis, drinking, phagocytosis, eating, receptor-mediated, a ligand bonds to a receptor, and it all three of them, once their thing is fulfilled, pulls it into the cell via a vesicle. Look at this, another set of slides that I love. If you know these two slides, you know the vast majority of what we just talked about. Go back through, make sure any of the stuff I highlighted in the slides is also known, but this will give you the big breakdown. Again, it sounds like a lot, but when you put it like this, it's not too bad. Yeah? Don't worry about the characteristics so much. Be able to basically summarize it. The big thing is give me an example of how it happens, okay? All right. That basically sums up the organelles and the cell transport systems. Next, we're going to talk about the cycle of the cell. This is the cellular reproduction. This is how we replicate cells, okay? So what I want you to know about the cell cycle is there are three main phases: the interphase, mitosis, and cytokinesis. Cytokinesis is a phrase which means the division of the cytoplasm. In cell reproduction, the cells that are created are called daughter cells. Okay? When we're performing cellular reproduction, we have checkpoints. There are different parts of the cell cycle, and as we progress through the different cell cycle portions, we hit checkpoints. A restriction checkpoint is basically one that says, hey, have we met the requirements to move on to the next step in the process? To either divide or not divide, or if something went wrong, to kill the cell so it doesn't become a problem or a misshaped cell. Yeah? Most cells do not divide endlessly. I mentioned this, I believe, in our second lecture. The number of times you can divide is based on the amount of DNA garbage you have at the end of your DNA, called telomeres. Every time you replicate your DNA, you don't perfectly replicate it, and you end up cutting off some of the DNA. Fortunately, when we're born, there's a lot of junk DNA thrown on the end of the good DNA called telomeres. As you replicate, you lose those telomeres. Eventually, you live long enough that as you replicate, you start destroying important parts of the DNA, which is why eventually our organs slow down, stop functioning, and eventually we die. So when we go into cellular reproduction, at each step of the phase, our cell goes through a checkpoint to say, hey, did we do that process correctly? Okay, we did, let's move on. And then at some point we get to a restriction checkpoint where it says, hey, we did the thing. Do we want to divide again, or are we done dividing? And oh, did we divide poorly and if this is a misshapen cell? Okay, it needs to die then. Yeah, that's what I want you to take away. The phases of the cycle are called the interphase, mitosis, and cytokinesis. And this is what it looks like. So first, we have our G1 phase. That's our interphase, G1S, G2. Don't get in the weeds with this. I don't really care about interphase. What I want you to know is that in interphase, when a cell is replicating, the interphase is when we copy everything. So if I have one cell and I want to turn it into two separate cells, well, first we're going to need to grow extra of everything so that we can divide. Then we need to copy the genetic material. Then we need to copy all the organelles. We need two of everything so that when we split the cell, we have two cells with one of each, yeah? At each one of these stops, we have a check to see, hey, did we accomplish it? With cell growth specifically, as we begin building things up, if there's a problem here, that is our restriction checkpoint. If everything looks good, we proceed to division. If we don't need to divide anymore, we just stop and we remain our own specialized cell. If something went wrong, we perform what's called apoptosis and we destroy the cell. We'll talk more about apoptosis later. What I want you to know here is that the first phase is interphase. It is called G1, S, and G2. G1 and G2 is when we duplicate all of the non-genetic material. S phase is when we duplicate the genetic material, and your restriction checkpoint is after G1. That's what I want you to know. Once we've done the interphase, then we perform the actual reproduction, the mitosis. And finally, we perform cytokinesis, where the massive cell that now has two of everything splits into two separate cells with one of everything. Mitosis has four phases: pro, meta, ana, telo. So, summing up what I already said, interphase, it's before division begins, in which the cell grows. We synthesize new molecules, new membranes, new organelles, new DNA. It is a time of great synthetic activity. A lot of stuff is growing. G1 and G2 we call gap in growth. I just want you to know it's when we're building everything that's not the DNA. S is when the DNA is replicated. So once we have replicated everything, what happens? Well, once we have a megacell that has a bunch of doubles of everything, we now need to divide into separate cells. There's two types of cellular division: meiosis and mitosis. You don't need to know meiosis. All I want you to know is that meiosis, an example of it is sperm and egg. Instead of fully replicating everything and dividing in such a way where you have a full new cell twice, meiosis doesn't fully replicate the DNA. It replicates everything else in the G1 and 2 phase, but it doesn't replicate the DNA in the S phase. And so when the cell divides, each daughter cell has half the genetic information. That is how sperm and egg cells are produced. So all I want you to know about meiosis is that sperm and egg cells are an example, and it results in half the genetic information in each daughter cell. The one we are going to spend time on is mitosis and cytokinesis. This is the vast majority of cell replication because it creates a total copy of a cell. So imagine if you get a cut in your skin, oh, we have damaged skin cells, we have a big hole in our skin, we need to fill that with new cells. Mitosis is the replication of those skin cells to fill that gap. So it increases the number of cells in growth and development, and it's involved in, of course, wound healing. Mitosis is the vast majority of cellular reproduction. Okay. In mitosis, we divide the nucleus and then we divide the cell. Okay. Each daughter now has an exact copy of the same DNA, a full copy. Meiosis is a half copy amongst the daughters. Mitosis is a total copy amongst the daughters. So there are the 4 phases. We have the prophase. What I want you to know here in the prophase, this is when the DNA, which is in its chromatin phase stage, condenses into much bigger DNA structures called chromosomes. Then the centrioles, remember the centrioles, they are made, 2 centrioles create a centrosome. They are going to start traveling to opposite sides of the cell and release microtubules, which are little arms that come out and attach onto the chromosomes. Okay? During this phase, in order to prepare the chromosomes to be divided, we have to disassemble the nucleolus and the nuclear membrane, also known as the nuclear envelope. Okay, so the things that enshrine our nucleus and our nucleolus, those membranes, have to dissolve. That way we can start pulling apart the DNA. So in the prophase, DNA condenses into chromosomes, centrioles migrate to the poles, and in order for them to make contact via microtubules with the chromosomes, we have to dissolve the nuclear envelope and the nucleolus. The metaphase. During the metaphase, those microtubules that the centrioles have created reach out and they attach to the chromosome. They attach at the center of the chromosomes because at this point the chromosomes are duplicated, so the 2 copies are attached to each other at what's called a centromere. Just understand a centromere means the center of, yeah? So the chromosomes are attached to by those little microtubules, and once they attach, they start lining the chromosomes up so they align along the mid portion of the cell. And so the centrioles release the microtubules, these little arms that attach to the chromosomes, and align them nice and orderly along the midpoint of the cell. Once we go through all these phases, I'm going to show you a picture. It's going to make a lot more sense. The anaphase. So, when a chromosome is copied, it's attached to one another at the centromere. When those microtubules attach to the centromere, they pull the chromosomes apart. So you now don't have 2 copies connected together. You have the free copies separate from each other. We call those chromatids. Okay? They are now individual copies of chromosomes that are separated. Yeah. So initially the chromosome is duplicated and it's and the duplicated the copies are connected to each other. When we rip them apart, they're called chromatids. Okay. Then the spindle fibers, right, those microtubules shorten and they pull the new chromosomes to the sides of the cell. So they're not lined up in the center anymore. They're starting to pull to the different sides of the cell. And then finally in our telophase, those chromosomes that have been pulled to opposite sides of the cell start to redevelop their own nuclear envelope and nucleolus. Now we have a cell with 2 nuclei. The chromosomes that are now in that nucleus unwind back into their chromatin state. Now they look like they did when they started. So let's look at this picture. We have a cell that's going to divide. So we go into our prophase. The nuclear envelope and the nucleolus dissolve. The chromatin condenses into chromosomes, and we can see that's 1 copy of a chromosome, that's another copy. They're connected at a centromere. The centrioles begin to migrate away from each other to the opposite sides of the cell, and they produce microtubules. During the end of the prophase, they are now on opposite sides. Those microtubules now connect to the centromere of those chromosome pairs. We call them sister chromatids as well. In the metaphase, those microtubules push and pull those chromosomes and line them up nice and even. So we can see now that the DNA, we have one chromosome here, one there. They also call them sister chromatids because it's the same thing copied. That's the metaphase. It aligns it nice and orderly in the middle. In the anaphase, these microtubules shorten and it breaks them in half. So now the sister chromatids are now separate chromosomes. These are copies of each other. 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 And those microtubules shorten until we get the genetic material condensing into each side of the cell. Finally, in the telophase, that condensed chromosomes form their own nuclear envelopes, their own nucleolus, and then they unwind and turn into chromatin again. So now we have one cell with two fully functioning nuclei. Remember, all of the stuff in here, all the organelles, all the cytoskeleton, they're also duplicated, but they don't get specifically divided evenly because we don't need them to be divided evenly. The cell can make more of whatever it needs to. But during the cytoplasm here, everything else is duplicated, so there's enough for both cells eventually. Finally, cytokinesis happens. Cytokinesis is simply the pinching of the cell membrane so as to break away from each other and form two new daughter cells. Okay? So again, we have the interphase where we are duplicating everything, double of everything in here. The prophase, the centrioles travel off to the sides to the different poles. The chromatin condenses into chromosomes. The nuclear envelope and the nucleolus dissolve their membranes so that they're now freely floating. The centrioles produce microtubules that connect to the sister chromatids, the duplicated chromosomes, at the center point called a centermere. Then, in the metaphase, the microtubules push and pull the chromosome and line them up nice and even. In the anaphase, those tubules shorten and they rip the sister chromatids apart, creating one copy of chromosome, two copy of chromosome. They pull off the chromosomes to the sides, where in the telophase, new nuclear envelopes and nucleoli form. The microtubules go away. The chromosomes unwind back into the chromatin. And then finally, cytokinesis. The membrane, the phospholipid bilayer, contracts and pinches and eventually breaks off from each other, forming two new daughter cells. At this point, the daughter cell will either go off and be a cell, will say, oh, I want to reproduce again, so I'm going to double up on everything and go here, or it'll hit a restriction checkpoint and say, something's wrong. We're going to destroy this daughter cell because something didn't go right. Something went wrong. So again, the cytokinesis phase is when we separate the cell membrane, okay? It technically begins in anaphase. As we start pulling the chromosome aside, that's technically when the membrane starts to create this contraction that we call a cleavage furrow. And it continues throughout the telophase until it pinches so tight that it tears and becomes two cells. The contractile ring is just the microfilaments that attach inside of the membrane. So the attachment points that cause this contraction is called a contractile ring, and it's made up of microfilaments. Yeah. Remember, microfilaments make up fibers of muscle. It has to do with movement, the movement of the cell membrane to contract. The two new cells might have slightly different amounts of cytoplasm and organelles, but they will share identical copies of DNA. And that's why I say there's a special process here to separate the DNA very evenly. But then within the cytoplasm, the amount of cytosol and organelles do double, but we're not as specifically worried that we get the exact same amount on both sides because we can always build more organelles. Okay. That's it. That's how it works. So again, we have the interphase. G1 and G2 is when we copy all the organelles and the cytoplasm. The S phase is when we copy the DNA. If something goes wrong at the end of the initial copying, we either stop the cell division and just go on, we continue with cell division, or if something truly goes wrong, we perform what's called apoptosis, which we'll get to. After we accomplish the interphase where we've doubled everything, then we attach to the chromosome, we move to the center, we pull it apart, we form new nuclei membranes, and during these last two phases, the cell membrane pinches off and forms two new daughter cells via cytokinesis. Cell creation. Okay, now that we have a cell, let's talk about differentiation. So a cell has a special function. All I want you to know is differentiation is the process by which the cell decides what kind of cell it is. Understand that every cell in the human body, in the nucleus of that cell, is the DNA for everything. So a lung cell in its nucleus has the DNA for a heart cell. It has the DNA for a brain cell. But when it decides to be a lung cell, it turns off all of the DNA related to being a heart cell, related to being a brain cell. And it only turns on the DNA information of being a lung cell. That process is called differentiation. It's choosing what kind of cell it is. Before we differentiate a cell, we have a special type of universal cell called a stem cell. You've heard of these before. A stem cell has no specialization. It is a cell that can freely divide as many times as it needs without specializing. So their presence allows for continuous growth and renewal in the body without it having to be specialized. So bones heal because bone cells create new bone cells. They're specialized. But if you need more help for a bone to heal, you can throw stem cells at it, which are not specialized. They can rapidly reproduce, filling the space, and then they can choose to specialize later into bone cells. So stem cells are great because they're kind of the cure-all. They're a type of cell that can become anything later on, and so they're known for self-renewal. Yeah, they can, the ability of a stem cell to divide and give rise to another stem cell is called self-renewal, and it can specialize later or differentiate later. A progenitor cell is a type of cell that is partially specialized. So a progenitor cell knows it's going to be a nerve cell, but it doesn't know what kind of nerve cell yet. It might be a brain cell, it might be a spinal cord cell, it might be a peripheral nerve cell. It's specialized enough to know that its genetic information that it's turning on is related to the nervous system, but it's not quite specialized enough to say where in the nervous system yet. So stem cells can become anything. Progenitor cells can become anything within a type. So a stem cell and a progenitor cell allows cells to retain the ability to reproduce and differentiate later. So when do we actually see these cells? Well, when sperm meets egg, we have a fertilized egg. As that fertilized egg reproduces in the early development of an embryo, those cells that are reproducing are all stem cells. They all contain all the genetic information of everything, but they haven't turned anything off or on yet. They're just stem cells. Then, as the stem cells become more numerous, they start to become progenitor cells. Some of the cells turn off the information for everything and turn on the information for skin. Where in the skin? We're not sure yet. Could be epithelial, could be a sweat gland. Or some of the progenitor cells will specialize to nervous tissue. Is it going to be an astrocyte? Is it going to be a neuron? We haven't decided yet, but we do know it's going to be a nervous tissue cell. So it's a progenitor nervous tissue or a nervous cell. Yeah. And then eventually the progenitors of all the different bigger categories will further specialize and say, okay, I am skin now, or okay, I am a sweat gland or a sebaceous gland. The nervous progenitors will now say, okay, I'm a neuron, or okay, I'm an astrocyte. All I want you to know about differentiation is by turning off most of the DNA information and turning on only some, that is how a cell differentiates. I want you to know that a stem cell is a cell that has no differentiation. I want you to know that a progenitor cell is a cell that has some differentiation, but not a lot yet. Finally, let's touch on apoptosis. I want you to know one thing. Apoptosis is cell death. If something happens incorrectly during reproduction of cells, the body will recognize it and it will say that cell was made wrong, cause it to die. It'll activate a signal. If you want to know what it looks like, the cell will swell up, the nuclear membrane will fall apart, the chromatin will condense into chromosomes, and enzymes will tear the chromosomes into pieces, and eventually the cell will explode into tiny pieces, and then the scavenger cells in our body will go and destroy those fragments, so the cell is totally gone. Apoptosis means cell death. It kills cells, whether they're overgrown, damaged, or just extra, okay? All I want you to know about apoptosis is it means cell death. It's how we get rid of extra tissue, damaged tissue, or overgrown tissue. In a practical sense, I'd like you to try and remember that apoptosis is the most important in preventing cancer. When we divide cells millions of times every single day. And every single day, we mess up sometimes. And those wrong-made cells are called cancer. And if those cells reproduce out of control, that's how cancer spreads. Our body has a natural way of fighting cancer. It's called apoptosis. It will recognize the cell is bad and it will trigger it to self-destruct. We get cancer every single day. Every single day a human develops cancer, but our body recognizes and kills it. So when patients are diagnosed with cancer, it's because somewhere in the process apoptosis failed or the body failed to recognize the need for apoptosis, and as a result, those cancerous cells continued to grow. All cancer is deformed cells. Okay, that was a lot. But if you go through it and you go through the slides I put there with the red boxes, that break everything down, it really isn't a lot. Get good at telling the story. You don't need to know all the minor details. I want you to be able to recognize the story, okay? If I say which portion of mitosis is described when I say the centrioles tear apart the sister chromatids and pull the chromosome to opposite sides of the cell, I want you to say anaphase. When I say which portion of mitosis occurs when the chromosomes are lined up along the midline of a cell, I want you to say metaphase. What is it called when the cleavage furrow in the cell membrane is formed around the contraction ring to create two new daughter cells? I want you to say that's describing cytokinesis. That's the kind of stuff I want you to know. If I describe it, I want you to name it. If I name it, I want you to be able to recognize the description. When it comes to the organelles, I want you to have a rudimentary understanding of what does the organelle do. The Golgi apparatus modifies proteins and lipids to then send out of the cell or to other parts of the cell. The endoplasmic reticulum produces proteins and produces and absorbs fats. The rough endoplasmic reticulum deals with the proteins, the smooth deals with the fats. The ribosomes synthesize proteins. I want you to know stuff like that, yeah? Focus on the highlighted things, focus on the things I've emphasized here in the video, and don't get bogged down with the weeds of all like the filaments and whatnot. I want you to know microfilaments made of actin play a role in fibers in muscle, microtubules made of tubulin and play a role in cell division. Intermediate filaments, I'm going to tell you right now, I'm not really going to ask about them. I guess know they exist and know that their size is somewhere in between the really big microtubules and the really, the really big filaments and the really small microtubules. Yeah. So I'm putting this up Wednesday night. We're going to be doing this lecture live tomorrow morning. So this will be the last time that I will be giving you the lecture first, and then you will be receiving the video. From here on out, we are going to be on the track of I will produce the video, you will watch it, and then in class, I will do a speed run through the lecture so that we can spend more time answering your questions and drilling the things that you might be confused on. Okay? With that in mind, I'm going to be producing the video for the next chapter over the weekend. There shouldn't be any more trouble with rewriting things. So for next week's class, I would like you to have this video and the associated assignment done, as well as when I post the next video over the weekend, I want you to watch that video and have the assignment ready for that class as well, so that when we go into the lecture, we're caught up and we're finally getting our flow of video, assignment, lecture to clean up any questions we might have. Okay. As far as the supplemental syllabus goes, once we are finally caught up, which will be next week, I'm going to lay out my plan for when we're going to do our first exam. The way I'm going to do things is prior to any exam, we're going to have a day where we're going to probably lecture for a little bit, and then we're going to go through a bunch of practice questions so that you can, one, get a feel of how I'm going to ask the question, and two, kind of test your knowledge. Okay. That's it. I look forward to seeing everyone in class tomorrow. Have a great night.